Our next speaker, first of all, I welcome Dr. Tommy Lewis from Oklahoma Medical Research Foundation. He's an assistant member of the Aging and Metabolism Research Program. He will have his talk about the role of mitochondrial fission in neural development and disease. Welcome to our webinar or virtual meeting.
Thank you for inviting me to share our work. Let me get this shared. Okay. Yeah. Thank you again for the kind invitation to share our work today about the role that mitochondrial fission plays in neuronal development and disease. It's always nice to present and share, especially during these times, and to hear what others are thinking about your work. So we appreciate any feedback. And I have to apologize up front a little bit. I'm recovering from a cold, so my voice sounds a little deeper than normal, and I tend to have some coughing, so I apologize if that crops up in there. But our lab is essentially interested in the role that mitochondria play in regulating compartmentalized metabolism or compartmentalized functions within cells. And the reason for this is that essentially these mitochondria are dynamic organelles that can move around the cell and form unique compartments within the cell. And this is because of their double membrane structure that we all know well, and its ability to allow them to create strong gradients from the cytoplasm to the matrix. And this is what drives many of the functions we think of when we think of the mitochondria, right? It drives oxidative phosphorylation and the ability to produce ATP, but also many other functions that we're interested in cellularly, including calcium homeostasis, lipid biosynthesis and processing, as well as just monitoring the environment of the cell and determining if the metabolic environment becomes too terrible, for lack of another word, to survive and initiates the apoptotic pathway. But this creates some challenges when we want to study these dynamic organelles. And so when we look at this, we think of three main challenges that we face. The first is that it is necessary to study these mitochondria in a physiological and metabolically relevant environment. And this is because they, of course, respond to what they sense in the environment and change their functional output. So if we're studying them in a non-physiological environment, probably they're going to reveal to us functions that might not be relevant in an actual physiological environment. And then the second one is that we have to have the ability to observe these dynamic organelles and changes in their function, because as you likely well know, they can move around, they can undergo fission-fusion events, and this changes the function that we can observe. And then finally, the third one that I think has been largely underappreciated to this point is that we have to have the ability to actually distinguish what we like to call mitochondrial subpopulations within the cell. And this is because, as been shown in many cell types now, but is classically known from skeletal muscle, there are multiple, or at least two unique subpopulations of mitochondria in these cells, the subsarcolemmal pool and the intramyofibrillar pool. And I think what's interesting about these two pools is that they're obviously localized to different parts of the cell, but when the cell receives the same stimulus or challenge, they respond differently, meaning that the functional output of these mitochondria is somehow different. And in order to understand how they're driving compartmentalization within a cell, we need to actually take into account that these populations exist. So how does our lab work to try to overcome these? Well, the first thing we do is we use the neuron as a model cell to study this. And this is because neurons are uniquely highly compartmentalized cells that allows their function to happen, right? So we have the receiving part of the neuron called the dendrites here, which are these shorter, stubbier processes with spines on them, which is the postsynaptic side of the synapse. And these are specifically built to receive information from cells surrounding this neuron. Then that information gets processed in the cell body, integrated and sent out through the output part of the cell, the axon, which is this single thin filament that reaches out and then branches all around to connect to the other cells in the dish to form this circuit-like structure that allows these neurons to communicate. And along the axon are unique subcompartments called presynaptic boutons, which is where the neurotransmitter is released to the next round of cells. And the reason that we use this as a model cell to look at mitochondrial compartmentalization is that actually the mitochondria in these two regions of the cell look very distinct. So in the dendrites, and shown here in this higher magnification, these mitochondria are highly elongated and more fused, and they also occupy, you can see, a high volume of this compartment. But out here in the axon, and shown here in higher magnification, the mitochondria are small punctate individual mitochondria, which are localized to specific points along the axon. And so this model cell allows us to easily look at and visualize these two distinct mitochondrial subpopulations in the cell and try to understand what role they are playing in the function of these two unique compartments in the neuron. And then the second challenge is to try to actually study these in a physiologically relevant situation. And to do that, we use a technique called in utero electroporation, which allows us to visualize neuronal development in vivo. And so essentially we expose the uterus of a mouse, and then around E15.5, when the embryos are around E15.5, we inject DNA into the lateral ventricles. This can be plasmid DNA that encodes either for a fluorescent reporter or can be shRNA to perform knockdown or a CRISPR construct to knock in or knock out something. And then we give it a small shock to electroporate the progenitor cells in that region. And what that does is by timing this and targeting the electroporation, we can target a specific population of cortical neurons in the brain called layer 2-3 cortical pyramidal neurons. And then we can follow either macro level events such as axonal development and branching, or we can look at a single cell level and look at mitochondria structure in a neuron that is developed in a physiological environment. And so our first kind of foray into this was to actually confirm that in vivo, we still see this subcompartmentalized mitochondria structure. So again, here we have the cell, this is the axon coming up here and dendrites up here. And you can see the mitochondria are highly elongated in the dendrites and punctate in the axon. So this is conserved in vivo, which was nice and important to see. But then we couple this with in vivo imaging techniques and also ex vivo imaging techniques to visualize mitochondrial dynamics and functional changes that are occurring in live animals, live mice. And essentially I've custom built setup with a 2-photon where we head fix the mouse under the objective, but they are running along this treadmill or walking along this treadmill. So we can, and then we install a cranial window in the skull so that we can image through that cranial window while the mouse is awake and either sitting on the treadmill or walking along that treadmill, so that we can visualize this in again, a physiological environment. So using this kind of study, our previous work has shown essentially that along the axon, mitochondria become less dynamic as the axon matures. So early in development, they are moving around and trying to find their position, but then they become captured at specific points along the axon, and their dynamics become much less as the axon matures. And this is through a specific kinase pathway, the LKB1 NUAC1 kinase pathway, that regulates the timing and the presynaptic capture of mitochondria. And this is really important for proper branching and development of the axon. But this didn't really get to sort of the main question that I and my lab are interested in. And that is, why do neurons need this two seemingly different populations of mitochondria? And basically, there are three main questions sort of that we are focused on trying to understand this. So again, why do they need this? Do these mitochondria imbue some specific properties to the axonal or dendritic compartment that shapes their function? Or is it required because of energy production issues or something along those lines? And then the second one is actually looking more at the mitochondrial level. What is actually unique about these mitochondria, these small versus large mitochondria? Do they have unique properties, or is it more that they just have the ability to provide more of some metabolite or more ATP or something along those lines? And then finally, more of like the long-term goal is to understand if this mitochondrial structure and function is disrupted in aging or neurodegeneration. So I hope I will share a little bit about each of these three questions today with you all. So to get to this first question, why are mitochondria small in the axon versus large in the dendrite? And what is the molecular mechanism that drives that? So we basically just made simple hypothesis based on observation, right? These mitochondria in the axon are small, so fission must be occurring more often than fusion to give rise to these small mitochondria, and the opposite must be true in the dendrites. So what regulates this increased fission along the axon? And then can we manipulate that to actually ask the question, why are they small along the axon? Now we actually know quite a bit about the molecular mechanisms that regulate this fission and fusion of mitochondria, and they are actually pretty highly conserved all the way from yeast to mammalian cells. And in many labs, including like the Nanari lab and such, have given us a lot of insight into this process. And so mitochondrial fusion, the process of joining two mitochondria together, is regulated by two GTPase families, MFN1 and 2 at the outer membrane and OPA1 at the inner membrane. And I think what's important to note about them is that they are mitochondrial proteins. They are kept on the membrane, either outer or inner membrane of the mitochondria. Whereas fission, the main effector, DRIP1, the GTPase that actually breaks the two mitochondria apart by undergoing scission, it actually is not a mitochondrial protein, making fission a little unique. It's a cytoplasmic protein, and then it's recruited to the outer membrane when necessary by a suite of receptors, MFF FIS1, and these MID 49 and 51 are also known as MIF1 and 2. And these are mitochondrial outer membrane proteins that have the ability to recruit DRP to the outer membrane of the mitochondria under certain situations. And so we made an observation and a hypothesis that if fission is high in the axon, that maybe the activity of one of these receptors is much higher in the axon than it is in the dendrites. And so to test this, we took a candidate approach and we developed shRNAs against some of these receptors, and in particular against MFF, or mitochondrial fission factor. And so we take in utero electroporation, we inject an shRNA plasmid, and we look at P21 at mitochondrial linked along the axon. And as you can see, as we have shown before, they're small and punctating control axons. But when we knock down MFF, maybe not too surprisingly, mitochondria get much longer because we've removed this fission factor, right? And this can be rescued by reintroduction of humanized MFF. So it's impervious to the mouse shRNA. And we can see nicely that this can be rescued, showing that this is actually targeting MFF. This is nice, but I guess what really got us excited is when we looked in the dendrites of those same neurons that we just looked at for the axons, we saw something that was a little more surprising, and that's that MFF knockdown seemed to have no actual effect on mitochondrial length or occupancy in these dendrites. So now it looks like we have a tool that shows that MFF is the main driving factor for this increased fission along the axon. And to show that this was the case, that we were actually disrupting mitochondrial fission and fusion and not having some other side effects or non-functional mitochondria or something, we used a photoconversion approach where we have MEOS2, which is a photoconvertible protein. It starts in this green state, and then upon activation with UV, we can photoconvert a small segment of the axon, and then we can perform time-lapse imaging and visualize new unconverted mitochondria coming into that region and look at fission fusion events as they occur in real time. And so you can see here a nice fusion and fission event. And so we did this in both the control axons and the MFF knockdown axons. And as you can see, there's a clear reduction in the number of fission events that occur along axons where MFF has been knocked down. But what I think the data really ended up showing to us and was kind of interesting for us is that in a control axon, what we see is that actually there are quite a number of fusion events that are occurring. But soon after that fusion event, a fission event occurs to break those apart, so they don't stay fused as long mitochondria. Whereas in the MFF condition, we see a fusion event, and that fusion event basically is never followed by the fission event, leading to this accumulation of longer mitochondria along the axon. So what's going on here? And is now that we have this tool that shows that MFF is the major player for fission in the axon, and it doesn't affect dendritic mitochondria, we can use this as a tool to test why mitochondria are small along the axon and what's happening here. So our first kind of thought was that maybe these mitochondria, this is like a structural issue. These mitochondria just can't get to the right place along the axon because they're too big to fit in the axon or too big to like move along the axon. Or they can move along the axon, but they can't get to the presynaptic batons or make a functional connection there. Clearly, this turned out to not be the case because when we did time-lapse imaging, these highly elongated mitochondria, they move along the axon perfectly fine. And when we look at their localization to the presynaptic site, it is actually kind of hard to compare because these elongated ones are actually overlapping many presynaptic sites. So if anything, it seems like they're more likely to be interacting with the presynaptic site, arguing that this isn't a structural issue, but maybe instead it's actually the functional output of these mitochondria that's important. So to try to understand how changing mitochondrial size changes the function along the axon, we started probing the different functional properties of the mitochondria that we could think of. The first one is membrane potential. We want to make sure that disrupting this fission fusion process isn't just like disrupting the total structure of the mitochondria, and they can no longer maintain these gradients that drive the function and make the membrane potential. So we use tetramethylrhodamine, which is a potentiometric dye that accumulates in the matrix of the mitochondria based on membrane potential. And so we give them a set amount of dye and a set amount of time, and we look at how much of that dye accumulates in the matrix. And what we could see is that these elongated mitochondria, they still accumulate tetramethylrhodamine at pretty much similar levels to the mitochondria. So they do maintain a membrane potential. And then we look at ATP production. As we all know, mitochondria are called the powerhouse of the cell, and that's what everyone likes to think of them as. So we wanted to check using a FRET probe called mito-18 that is sensitive to ATP levels. We target that to the matrix of the mitochondria, and then we can calculate FRET, or the YFP to CFP ratio, and we can look at basically relative concentrations of ATP in the matrix. And we see that the relative concentration, or the relative FRET, is similar in these mitochondria, suggesting that they have similar levels of ATP in them. And then to make sure that they still are kind of responding in similar ways, we also did some challenges to these mitochondria where we treat with antimycin A, and we look at what happens to either membrane potential or the pH of the matrix, which we would expect when we shut down the electron transport chain to be affected, right? And so basically they have similar sort of baseline and similar change in fluorescent intensity for both probes when we treat them, meaning that they seem to be responding pretty similarly to the same challenge in this case. So what is going on? And we started to get some clue about this when we started to think about the calcium handling ability of the mitochondria. So Dr. Kwan had shown previously in my postdoctoral lab from Peru that basically we know that when the action potential comes down the axon, it triggers the opening of voltage-gated calcium channels in the membrane, and this leads to an influx of calcium into the presynaptic site. This influx of calcium is what triggers the exocytosis of neurotransmitter from that presynaptic site. But what Dr. Kwan had recently shown is that at presynaptic sites that contain a mitochondria, actually much of this calcium that floods in is quickly imported into the matrix of the mitochondria in a mitochondrial calcium uniporter dependent manner. We don't exactly know why it's taken in there, if it's to stimulate metabolic reactions in the matrix of the mitochondria, or if it's to mainly buffer it from the presynaptic site. But essentially what it does is buffer the calcium that's available to trigger this release. And so paradoxically, presynaptic sites that you would think would release the most synaptic vesicles or neurotransmitter, because they have ATP, plenty of ATP there from the mitochondria, they actually have lower release because this calcium is not available to trigger exocytosis. And so this got us thinking that maybe these highly elongated mitochondria are basically acting as a calcium sink and just sucking up so much calcium that it blocks neurotransmitter release along the axon. So we set out to test this using a number of fluorescent probes that are sensitive to calcium. And so we have triple labeling. We have the labeling for the mitochondria, and you can see this nice highly elongated mitochondria. Then we have labeling for the presynaptic site, so we know where the presynaptic connection is being made. And then we have GCAMP, which is a calcium sensitive fluorescent probe that's targeted to the mitochondrial matrix. And then we can stimulate these axons with a set number of action potentials at a set frequency and visualize if calcium uptake occurs. And so we could visualize that clearly upon this activity, the mitochondrial matrix fills with calcium. I think it's important to mention that it still starts from the presynaptic site, so they've maintained functional connection with the presynaptic site. And then when we look at the integrated calcium levels taken up at the same activity for either these small mitochondria in the wild type condition or these elongated ones, we see that they are taking up approximately 3 times more calcium at the same activity levels for the axon. And so now this started to make sense to us because we could predict that by sucking up too much calcium under these activity conditions, they basically are reducing the amount of calcium available to trigger neurotransmitter release. And we can see here that when we target the GCAMP to the presynaptic space using VGLUT1, that we get less accumulation of cytosolic calcium at the presynaptic site. And then when we visualize neurotransmitter release using the synaptophysin fluorine, which is a pH-sensitive probe that when the vesicles fuse with the membrane, so normally they're acidic, but then when they fuse, they become more basic. The environment becomes more basic and allows that fluorescence to become greater. So we can visualize the actual exocytosis events, that we get about a 50% reduction in the number of synaptic vesicles that fuse with the membrane under these conditions. So what role does this play in axonal development? Well, we know that activity-dependent branching is an important part of stabilizing axonal connections in the brain. And so when we look using in utero electroporation at the development of these axons, what we can see is that the neuron cell bodies are here and that they project their axons across the corpus callosum to the contralateral side of the brain. And then they form their connections with layer 2, 3, and 5 neurons on the opposite side of the brain. But when we knock down MFF, while these neurons still form an axon and they send that axon to the contralateral side, you can even see that it tries to make the connections and invade the upper layers, that we get a great reduction in the branching or the stabilization of the connections that occur on the side of the brain. And this can be rescued by, again, reintroducing the human MFF that's impervious to our shRNA. So showing that this is really mediated by keeping axonal mitochondria at this very precisely regulated size along the axon, and disruption of that really disrupts axonal development. And so essentially, we think that by maintaining the size of presynaptic mitochondria via an MFF-mediated fission event, it determines the capacity to basically regulate calcium and provide a mechanism for tuning circuit connectivity and axonal development. But as you can imagine, this leaves a number of big questions that we are still trying to address. And the main one being still, how is MFF only regulating fission in the axon and the dendrite? Fortunately, I don't have a full answer for you today because it's something the lab is working on. But our first hypothesis, which we were hoping for because it would have been easiest to explain, is that MFF is only found on axonal mitochondria and not on dendritic mitochondria and neurons. But this turns out to not be the case. If we look at our labeled neuron here, we can see the dendrites here and the axonal mitochondria here. And then we stain for endogenous MFF, and we were lucky that there actually is a good antibody that recognizes mouse MFF. We can clearly see that these dendritic mitochondria are decorated with MFF. So there is a pool of MFF in the dendritic mitochondria, but it seems to be in some sort of turned off or inactivated state. And we're still working to understand how exactly this occurs. We have some thoughts about this based on the work from Jian Zhao and Monica Nestor's lab at the Karolinska Institute, where they showed that actually these MFF and other fission factors, MIF1-2, they create unique complexes. And this changes the ability of MFF to recruit DRIP1 to the mitochondrial surface. So we have been developing shRNA constructs to knock down each of these other fission factors, as well as trying to actually visualize their localization this has turned out to be quite challenging because while people have studied MIF1 and 2 function in many sort of human cancer cell lines, almost no one has studied them in mouse ones. And so none of the antibodies are good, and none of the published shRNA sequences are against mouse proteins. So we're developing them all kind of along the way. But at the same time, we also are trying to implement this new tool using this orange construct published by the McGillivray lab to basically knock in GFP to the endogenous locus for each of these receptors. And they've designed this really interesting construct that has basically all the elements you need on the same plasmid. So you have your guide RNA, your tag with the homologous arms, as well as the Cas9, so that we can use this in neuron systems. And we have been screening these in NIH3T3, a mouse cell line recently, and we're really excited. We finally have first guide for MIF1 that seems to work well. We can see nice punctate localization of this MIF1 on the mitochondrial thing. So I think this will be a really critical and powerful tool for us, because not only does it allow us to visualize the localization and abundance, the endogenous abundance of these mitochondria in different mitochondrial pools, but it allows us to perform live imaging and watch fission fusion events that are occurring in these different regions and actually visualize which receptors are involved in those events. So I hope that soon we will have some interesting data regarding this to share with you all. So now I want to switch, I guess, lanes a little bit, but really this inactive pool of dendritic MFF has got us thinking about this kind of in a different light, and really wondering basically if having this inactive pool of MFF there is maybe a source of some of the problems we see in aging or neurodegeneration, leading to increased mitochondrial fission. And basically, is this pool somehow incorrectly activated as neurons age or are insulted by neurodegenerative processes? So I'm going to share with you a unpublished story that we have been working on. And it basically is looking at an Alzheimer's disease model and asking if MFF might play a role in this. And so a little background, we all know about Alzheimer's disease, that it's a devastating disease that leads to cognitive decline early on and then progresses to more severe phenotypes of movement and mood disorders, and also finally to death, right? And there are two major clinical sort of post-mortem markers that they look for to identify these things, and that's APP processed A beta 42 or tau, which forms either A beta plaques, extracellular plaques, or tau, which forms interneuron tangles. So I'm not going to talk too much about the tau side of things today, but instead focus on this A beta 42. And that is because many labs, again, have been working on this, and mainly the Malinal lab at UCSD was one of the first to show that actually before any of these plaques or tangles form and before there's major like symptomatic things, there's an accumulation of these A beta 42 oligomers in the milieu, the extracellular milieu, and that this extra A beta 42 oligomers, they start to bind to the surface of the neurons through some unknown mechanism. But when this happens, it basically triggers activation of different receptors in the postsynaptic side, the NMDAR receptors and voltage-gated calcium channels. And this leads to a massive influx of calcium into the neuron. As you can imagine, this is fairly important that calcium be highly controlled in the neurons because, as I showed you, it regulates axonal neurotransmitter release, but it also regulates action potential activation threshold and many parameters for the cell. And in my former postdoctoral advisor's lab, Dr. Poulou's lab, they had started to follow up on this, and they identified that there was this specific stress response kinase pathway that becomes activated under these conditions called the CAMKK2 AMPK pathway, where this extra calcium activates CAMKK2, and this CAMKK2 then phosphorylates AMPK, activating the AMPK complex. But as you may know, if you worked on or looked at AMPK at all, there are many downstream effectors of AMPK. And what was actually happening downstream of this AMPK activation is still largely, I would say, unresolved at this point. But we happen to have the fortunate interaction with Ruben Shaw's group at the Salk Institute. And at that time, they had been working on this project to look at AMPK and downstream effectors. And they identified that AMPK directly phosphorylates MFF when activated, leading to mitochondrial fission and eventually to mitophagy in cell lines. And so essentially we want to ask, is this AMPK activation then activating this inactive MFF that's decorating dendritic mitochondria, and if this plays a role in the phenotypes that we see? So to do this, we set up a culture-based system to try to do this in vitro, where we grow cultured neurons. We label them by electroporation so that we can visualize their morphology and mitochondria, and then we treat with these A-beta 42 oligomers. And many labs have already shown this, that when you treat with these A-beta 42 oligomers, the spines start to disappear along these dendritic processes. And this is because they lose their connection to the other neurons that are surrounding them. But we visualize that this coincides with the massive remodeling of the dendritic mitochondrial network, where they become highly fragmented and more individual-like mitochondria, more like axonal mitochondria. And so this got us kind of interested in maybe this plays a role in this process. And so to begin to sort of diagnose this, we were again fortunate to have access to a double flocked AMPK alpha 1 and 2 mouse line. So the AMPK alpha 1 and 2 subunits are both flocks. So if we don't provide CRE, basically it's like wild type mouse. But when you give CRE, you can remove both the AMPK alpha 1 and 2, and this leads to basically loss of the whole AMPK complex. So as we've already shown in control or CRE negative neurons, basically treatment with A-beta 42 again leads to loss of spines and fragmentation of the mitochondrial network. But when we knock out AMPK by expressing CRE in these neurons, we got really exciting and I think interesting result where these mitochondria no longer become fragmented, but also it leads to maintenance of the spines along these dendrites, suggesting that potentially this can be a causative linkage between the fragmentation of mitochondria, loss of mitochondrial function, and this loss of spine maintenance. And so this showed that it is in fact important that AMPK be activated and is playing a role in this process. But as you can imagine, just like blocking AMPK may not be a super good strategy. So we wanted to dig further if this is mediated through this interaction with MFF. And so we took, again, our neuron cultures, and we have our control shRNA, or we have our MFF knockdown experience. And you can see that when we knock down MFF, again, this rescues mitochondrial fragmentation phenotype and rescues the spine loss phenotype. So this got us excited, but as I previously showed you, knocking down MFF all over the cell is probably not a good strategy either, right? Because this probably has negative impacts on the axon, even if it's having a positive impact in the dendrite. So can we find a way to look at this more specifically by focusing on the phosphorylation by AMPK? And so to study if this is specific to the AMPK phosphorylation, we use what we call a replacement strategy experiment, where we knock down the endogenous MFF, mouse MFF, and then we replace with either human wild type MFF or human MFF, where different serines have been replaced by alanine to block this phosphorylation by AMPK. And as you would expect, returning wild type MFF, you get fragmentation and loss of spines. But when we found two serine sites that seem to be relatively important in this process, because when we mutate them to alanines, it's serine 155 and 172 on MFF, that this no longer led to this fragmentation or loss of spine phenotype. So this is really, I think, important for us, because what I haven't shown or told you yet is that it's already been published that loss of AMPK has no effect on axonal development, and we checked, it also has no effect on axonal mitochondrial size under normal condition. So this really seems to be a stress response sort of condition or pathway that's activated. And normal situations, it doesn't really need this to maintain normal development. But in this stressful condition, when it becomes overactivated, leads to this fragmentation. And so this might be a sort of targetable thing that we can target just this AMPK-based activation of MFF and prevent this. So next, that was all in culture. We want to actually see, is this something that's happening in Alzheimer's disease mouse models? So we are using this APP overexpressing J20 mouse model. This is a humanized, a mutated APP, and it expresses at a pretty high level in this mouse model. And so this leads to high processing of the humanized APP to form these A beta 42 oligomers. You can see this nicely in the hippocampus. And already at 3 months of age, they have significant spine loss. And so we can use this to look, and using, again, in utero electroporation, we could label mitochondria in these two regions, and we see this pretty strong fragmentation phenotype already at 3 months and loss of spine in these dendrites. And so we think this recapitulates what we saw in vitro. And of course, we still have a lot of work to do to try some of these rescue experiments and our rescue constructs in vivo in these mouse models. And of course, we would also like to look at other mouse models of Alzheimer's disease. As you guys may know, there is no actual really great mouse model of Alzheimer's disease, but there are multiple models that we could check this in and try to see if this is sort of a universal feature of many of these models. And so we have this preliminary sort of working model that under normal conditions, there is some calcium influx into the postsynaptic site with neuronal activity, but this is carefully buffered and monitored so that this stress response pathway is not activated or is very lowly activated for spatially, temporally regulated events. And we have these elongated mitochondria that take up calcium and play an important role in this calcium homeostasis. But in these sort of Alzheimer's disease-like model, where we have this A beta 42 driving this increased calcium. This leads to this stress response pathway being highly activated, the fragmentation of the mitochondria and potentially lost, because we also know that AMPK interacts with the autophagy machinery, leading to these smaller, less efficient mitochondria, or even fewer mitochondria. And this probably exacerbates this calcium response, because normally they would play a role in this calcium homeostasis, but now that's lost. And then linking this to this synaptic loss phenotype that we see. And so that's kind of where we're at with this. So I'll just go ahead and acknowledge my lab members. We're still fairly small, but growing, and my collaborators, especially Frank Blue, my postdoctoral advisor before I started my lab here, and our funding source. And so with that, I'm happy to take any questions that you all might have.
Thank you for your very interesting talk. I don't know if any of the participants have any question. Can you raise your hand if you have any questions so we can open up your microphone? Well, let me ask a question myself, okay?
Sure.
You talked about the role of the calcium in the mitochondrial fission. If I use any calcium channel blocker, such as verapamil, would that interrupt the mitochondrial function or in Alzheimer?
It's kind of hard to say. So I think it doesn't play a huge role in the actual fission-fusion process itself. But it may, lead to activation or something of these. So it's a calcium channel blocker, you said?
Yes, calcium channel blocker. Because you said for this function of the mitochondria, you need a calcium to get inside. And it came out of my head that if I use a calcium channel blocker, or even if you can find out people who are taking calcium channel blocker, would they have Alzheimer delayed or not?
Yeah, I'm not sure. That's a great question. My prediction would be that it should have a potentially positive effect, in delaying like some Alzheimer's like disease, but we have not tried any experiments like that yet. But that's definitely something we will be thinking about carefully
now. Okay, thank you. I think Dr. Mirina Najastadeh, yes, can you talk? Yes.
Okay, thank you. Thank you, Professor, for your very, very nice lecture. I really enjoyed it. Thank you. And my question is about the mitochondria fragmentation you showed in your last slides. You explained about them in Alzheimer's disease. And my question is that, is this fragmentation because of fragmentation and neuronal loss? I mean, at first the neural loss occur, and then because of the neural death, all parts of the neuron will be defragmented, and the mitochondria also may be destroyed. Not exactly only mitochondria. I mean, the whole cell may be fragmented.
Yeah.
What's
that's an important question. I didn't explain that well. So we think this is occurring early, much before the neuronal death. So this is occurring way before like the plaques or the tangles actually arrive. This is occurring when just these A beta 42 oligomers are in there. Now it might be occurring at a similar time point when they're starting to experience like some increased excitability or maybe excitotoxicity, but the neuron themselves, like I don't know if you could see on the screen, but they look relatively healthy and intact still at this point. So we think that this fragmentation of the mitochondria is occurring early before actually the neuron has started to initiate like apoptosis or anything like that.
One of our colleagues have one question in this. Professor Georgiani, can you raise your question?
Thank you for your interesting talk. You said that the mitochondria differs between the axon and dendrites. My question is, what's the difference between the mitochondria in axon and dendrite? What makes the specificity for the axon, the function, the related function of axon and dendrite? This differentiation between mitochondria in two sections of the cells.
Yeah, so that's a great question.
My question is clear. Is clear my question?
So you're asking basically what different functions do the two populations of mitochondria have in the two parts of the cell?
Yes.
Yes, the difference between the mitochondria in axons and dendrites. This difference, how related to the function of axons and dendrites?
Yeah, so this is a difficult question for us still to answer, because the real answer is we don't understand it completely. One thing that is been pretty well shown, I think now, is that their calcium buffering capacity is much different. And so, as you may be familiar with, in the axon and the dendrite, calcium, in the whole neuron, the calcium homeostasis is critical because it regulates all of the activity processes that are occurring in both the dendrite and the axon. And so there's some work, I'm totally forgetting the name of this lab at the University of Maryland. They showed that these elongated mitochondria in the dendrite are critical for long-term potentiation in the dendrite, in the postsynaptic side. And we've shown that this calcium buffering properties are critical for presynaptic release along the axon. So this calcium buffering is one of the main things that we've been able to show so far. What we still don't understand, I would say, and have a very good handle on, is what role they play in different metabolic processes. And the other critical thing that we know mitochondria do, but we really don't understand very well yet, is the lipid processing that occurs in these mitochondria. Because if they are processing different lipids distinctly in the axon versus the dendrite, this could play a critical role in making the membrane properties that give excitability and regulate excitability in those different compartments.
Thanks.
Okay, I don't know if you can see the chat. There is a question on the chat box. If you don't see it, I can read it for you.
Let's see, I think I can see it. So this is the one about a drug for...
If you can find a drug that can work on the mitochondria, would be that help the Alzheimer. That's what I understand from that question.
I think, this is $1 million question or $1 billion question. There are, I know, a couple of pharmaceutical companies who have invested a lot in this. I think that would be a good direction to look in, but we don't know. we know that the pharmaceutical companies invested a lot in these A beta removing drugs, and that turned out to not play such an important role in the human condition. So, I mean, that is one big caveat so far to all of the work in the Alzheimer's disease field is translatability from the mouse models to human actual physiology has not been particularly promising or helpful yet.
Okay, thank you. I don't see any more question. Thank you very much for accepting our invitation to have a talk in our Congress. I hope we will see you again for the future Congress that we have in pharmacology and physiology.
Sure, thank you for the invitation.
Thank you. خیلی ممنون از همه حضاری که تا این ساعت با ما بودن. همونطور که میدونید ما مجمع عمومی بلافاصله بعد از این سخنرانی خواهیم داشت. حتما به همکارانی که الان وصل هستن در واقع مجمع عمومی الان شروع میشه.